wanted to say a few words on, some of the things that you can do with the RETeval device that, since this is kind of a research market, I've tried to couch my terms a lot in of what kind of special stuff you can do, but just a small agenda of, what do you get out of the box before I try to get into kind of the custom protocols. And then, for custom protocols, it also depends on what, the device's capabilities and limits are. And then I'll touch on some of the PC tools that you can use to help make your life easier. So the RETeval device can be used for the ERG, which is what they all talked about, the other three speakers. It could also be used for a flash VEP where you record from the back of the head. Randy mentioned that it can be used as a pupillometer. Just to make it clear on one of Randy's points was that the device can measure the pupil size while it's recording the ERGs, because it does that to compensate for the pupil size, and it records those measurements. But if you want to do your own research on what the eye is doing during it, you can't simultaneously record video and record ERGs. So you have to, if you're interested in recording videos, then you have to do those at a separate time than recording the ERGs. And it can be used and has been used on both humans of all ages. I think the smallest kid's been maybe 2 kilograms that's done it, and then all the way up to the elderly. And it's been used on rabbits and dogs and pigs and monkeys. So, the device has an eye cup that kind of fits around the eye. There's a nicely placed sensor strip electrode. Besides doing the sensor strip, it also could come with a different adapter cable, so you can use any DIN electrode, and the device will even ask you what electrode type you're using, so that when we get normative data like Omar's study, and we're working on our own study of normal data in there, that can be tailored per electrode type. The device can do, the dark adapted ERG testing, the ICEV standard. It can do the light adapted testing, the flash and the flicker. These are the two that Omar talked about. Randy and Steve, one of the things they did was this photopic negative response, which had the red light on a blue background. It can also do on-off responses, so here the light stays on for a long time and then turns off, so you can get a separate response from the on bipolar cells and the off bipolar cells. And this is the way that Moorfields likes to do it, and this is the way that, from looking at kind of the research that Mineo Kondo and John Robeson and Laura Frischmann put together, what they thought was the best way. It can do an S-cone response, and it can do the flash VEP. So those are kind of the built-in, out-of-the-box capabilities. Custom protocols, so all the protocols, including the built-in ones, are actually used in a programming language called Lua. And having the protocols in a programming language has some nice features to it that's hard to get without a programming language. You can do randomization. So I gave a talk on the effect of flash duration of the photopic flash VEP at the I7 at ARVO meeting, and so I was changing the flash duration and looking at the response from different parameters, and by randomizing the order of the flash durations in the presentation, then the results here are this is a real effect. This isn't that these were done first and those were done later. Omar is since he's found that right-left difference, I gave him a randomized protocol where the device randomly chooses which eye to go first to try to help tease out, is it a second eye effect, or is it a left eye effect. And then if it's a left eye effect, well, then it might be electrode placement from your right-handed people. The programming language also lets you do reflex testing. Our diabetic retinopathy assessment protocol does some testing and then thinks for a second and says, I need to do more testing because there's too much variability. Or it could say, I'm good, I'm done. So that lets you kind of take just the right amount of data. You can also have the ability to call a protocol from a protocol. So you can say, like in our study to get normal data, we wanted to try to test all the photopic testing all the photopic test protocols that we have. So we just wrote a protocol that calls all the other ones rather than trying to write one master one. With a custom protocol, you can, the device will let you do these waveform review screens where you can select or delete different values. Of course, being able to select or delete values slows down the testing. So like in this study, I said, I'm doing everything in triplicate. I don't care what the data is. So then, here, because of I'm doing everything in triplicate, I don't care what the data is, the delay between tests was only 1 second, right? But if you have to show a waveform review screen and you have to decide whether you like it, then it's going to be more like, 10 seconds per test. So those are the kind of choices you can have with a custom protocol. In terms of the access to what can you do with the hardware, there's lots of ways to specify the stimulus. You can compensate for pupil size, or you can not compensate for pupil size. You can say what the color is based on the CIE chromaticity values, like white's 0.33 by 0.33, or you can say, no, tell me what the red's doing, red LED's doing, what the blue LED's doing, what the green LED's doing. You can compensate for the Stiles-Crawford effect if you're trying to measure pupil size. You can do automated timing. In this test here, there was a bright light, 6,000 candela per meter squared, that was on for like a minute with a dilated subject, and then you immediately start doing flicker tests afterwards. And the device timed, every 30 seconds, I'm going to do another flicker test. So you can try to see, what's the, how long does it take the eye to recover from a photo stress. So that kind of built-in timing can also be done. In terms of other stimuli that you can do these on-off protocols, leaves the light on for a while, then off, and off could not be off, but to a background level. The ringing in here is actually due to the post-processing filtering that happens. If you're interested in post-processing filtering, you can see that it rings before and after, so there's a time machine in there to know that there's going to have a response. That's called having a zero-phase filter, which preserves timing across filtering methods. You can have ramps where you step on the light and then you slowly take off the light and then you can step it on again. So you can look for kind of what's the effect of a ramp, of a step on, and then you can kind of fake the eye until I'm not really changing the light level and go down. Or you can have the opposite where you drop the light level and then you step it down and go up. You can generate sinusoidal light waveforms. And our sinusoidal light waveforms have really good, it's a really pure sine wave. There's the dashed line, the little dots are the best fitting sine wave, and the black line is the waveform. This is from looking at a photodiode's response, and they fall right on top of each other. You can do a harmonic analysis, and you can see that each harmonic is less than 1% harmonic distortion. So these sinusoids could be useful for, at a flickering rate like 28.3 hertz or 32 hertz. It can try to, any nonlinearities in the response are not coming from the photoreceptors, because their photoreceptors have a very linear response, so you can possibly get interretina function out of it. You could have a very slow sine wave, like a half hertz, 1 hertz kind of thing, to look kind of for how the pupil is changing. with brightness. So those are kind of the stimuli that you can do. In terms of the device capabilities, the device has ridiculously accurate timing. So as Omar said, the flicker implicit time was the most accurate thing. These are all production histograms from the last 659 devices that we've made, where we look at the error in implicit time for every device, and this is our pass-fail criteria is 0.2 milliseconds, and our actual standard deviation of devices is 1 microsecond. So the device only has one oscillator in it that drives the microprocessor, the ADD converter, and the stimulus generator. It's all done by the same clock crystals, so there's really no possible place for timing skew. In terms of the dimmest flash that you can make, our specification is 0.002 candela seconds per meter squared. This is our production data, so the worst case is actually 0.001. So if you want to do a little more than our specification, you can do a little more than our specification. Similarly, on the bright side, our specification is 30 candela second per meter squared. The worst case device we've ever made is 42 candela second per meter squared. So that's kind of the range of stimuli that you can generate with the device. And these two are with white light. So if you had only the blue light, then it will be dimmer on both the dim side and the bright side. In terms of PC tools, the device lets you. The device has a joystick control, but if you want to enter in something complicated for the patient name, it's a little bit of a drag. Or if you're writing in Japanese characters, it's even harder. There actually are Japanese characters in the device, but it's even a little bit harder. But you can type in patient information onto your computer, and then there's a way to scan a barcode that's generated on the device, on the computer screen into the device to enter in that information. And the data barcode app lets you enter in the normal stuff, patient ID and birthday. But if you want to add in extra stuff, it can be added. The device won't do anything with it other than pass it through. But you can enter in extra stuff if you want to have an encounter ID and other information. We have the device generates this raw data file that might be 10, 20 megabytes big, and in that raw data file it includes things like images captured directly after start test. Oops. It also captures a video before you hit start test, because like Randy said, we can't record video simultaneously with electrode data. But you can also try to get an idea of patient compliance or things like that. Geez, this was a pediatric kid and I got no amplitude, and you look at the photo and they're kids like this, and you go, okay, well, that's why there's no amplitude. That eyelid's clamped shut. So that raw data file viewer RIF browser is actually the thing that we use in the development of the device, so we aren't hiding anything back. It has all the raw ADD values, it has all the configuration of all the components in the device, so it has every possible bit of information. There's nothing else that's not in there. There's also a couple command line utilities for those that know a little bit of programming for programmatically extracting some of that raw data out. The numerical values you can get out by calling a RFF command line program. That's how I analyze data. I grab raw data files and I use the RFF program out of a shell. I use Mathematica to use and then drop to a shell script in it to call the command, grab the data, and bring it back into Mathematica. It also works with MATLAB. RIF image is how you get like these photos out if you don't want to use the raw data viewer, if you want to do it programmatically. I guess I'll end on John Lyon's comment. He asked, well, geez, you're showing statistically significant difference in the mean, but what happens if I'm interested in a patient, right? Then I need a better separation than a mean separation. I need to be able to say, what's the chance of this guy being good or not? And I think the answer to it is, like most things, there's a lot of variability among people. Like Omar said, there's 77% genetic variations in the ERG data. But I think one of the ways around that is to combine more information. Usually you have a milieu of clinical data before you get to make your final impression of a diagnosis. And one of the things that you can do in a statistical fashion to help do that is called logistic regression. And with logistic regression, you can try to combine metrics to improve the discrimination for these things. We actually did that in our diabetic retinopathy. There is a diabetic retinopathy assessment protocol in the device where it'll tell you, is this person at risk for vision-threatening retinopathy? And if your score is normal, you have a 99% chance of being okay. For Randy's optic neuropathy data, if you look at kind of two parameters, like Randy said, you can get this pretty good 85% sensitivity and 85% specificity for your individual subject, which is pretty good. With Steve's mental health data, you can also do kind of the same things. That he showed pretty good statistical differences in means, but the population still had some overlap. If you do a two-parameter fit, then you can get an area under the curve of 88%. So you can try to do some combinations of the devices, of the things, and I think kind of that's the way to try to make ERG data more definitive in a diagnosis, is to try to combine multiple ERG parameters, because they're generally, each ERG test is is emphasizing the results from different cells in the retina. But a disease could affect more than just one type of cell. It could affect lots of different types of cells. So by combining that information, you're able to make a better diagnosis. Anyway, that's all the comments I had. If anyone has any questions for me, I'll take some questions. Wow, I answered every question. I was that opaque. Great. Well, I really appreciate your guys' your time and attention, and I really want to thank our speakers, Steve and Randy and Omar, for all the for the great science that we were able to learn tonight, and their efforts in trying to make ERGs diagnostically useful for a variety of diseases to make everybody healthier. So thanks a lot for the speaking for us.